Explain disaster victim identification DNA infrastructure procedure. Every forensic DNA laboratory should make an effort to contact the relevant authority dealing with the emergency response and establish involvement in a possible fatality preparedness plan. Policy decision about people or about a sample collection, scope, and final goals of the effort will affect the victims' families and work stream and should be decided as early as possible. Internal response plan need to be addressed turnout capacity, sample tracking. Must have the names of supervisors responsible for different tasks that are updated as personal changes. Several sample types of for DNA testing should be taken at the earliest possible stage of the investigation provided traceability is guaranteed. Samples must be collected from each body or recognizable body part. Even if the identity is already established, a proper storage must also be assured. A single accurate reported missing list is of a crucial importance for streamlining the DNA identification process. Any submission of personal effect or family swabs need to be subsumed under a single case number. If multiple agencies or companies share sample collection or testing, the case number should remain constant. Multiple direct reference and samples from a first-degree relative should be collected for each missing person. Scientists with a background in genetics should be available for training or for consultation in the family liaison group. Disaster victim identification DNA testing should be only performed by laboratories with a demonstrated successful capability and continuous experience with these specified sample types. The set of loci to be analyzed has to be identified as soon as possible in coordination with the scientific community in the countries mostly involved. Minimum of 12 independent loci should be selected as a standard set, but even greater number of loci are preferred. All allele calls and all candidates matches are reviewed thoroughly. Composite DNA profile can be generated if derived from the same specimen or consistent for overlapping loci. The duplication policy should consider the logistic and circumstances of the mass fatality incident. If the standard autosomal STR typing fails to give sufficient information, adding typing such as mitochondrial DNA, Y chromosomal STR, or SNP markers may be used in a selected cases. A centralized database required for all data comparison. Electronic upload is recommended to avoid transcription error. Especially if multiple family members are involved, DNA-based identification would whenever possibly be anchored by anthropological or circumstantial data, a second identification modality or multiple DNA references. In disaster victim identification work, DNA statistics are best represented as a likelihood ratio that permit DNA results to be combined among multiple genetic systems or with other DNA device. Likelihood ratio threshold should be determined or for when DNA data alone can suffice for an identification. This will be based on the size and circumstances, closer and version open of the event. All evidence or circumstances should be checked in making identification, even if DNA provides the primary or sole identifying factor. The preparedness plan for the laboratory needs to include a policy for family notification, long-term sample deposition, and data archiving. Thank you.